Okay, Luke Bench here. Welcome to the December 2019 SNOC webinar, Developing Molecular Assays to Get the Dirt on Nitrogen Fixation, brought to you by New South Wales DPI Soils Unit, presented by Ashlea Webster. Ashlea is a microbiologist with New South Wales DPI Soil and Water R&D as part of the Australian Inoculants Research Group, or AIRG, which focuses on quality assurance and research surrounding rhizobial inoculants for legumes. Much of Ashlea's previous work, ranging from human bacteriology to veterinary virology to plant pathology, has involved working with pathogens. However, since joining the AIRG team, she's found working with beneficial microbes to be very rewarding. Ashlea has a passion for learning and aspires to unlock the mysteries held by rhizobia, and as such has taken the opportunity presented by her recent Science and Innovation Award to broaden her research interests into genomics and genome sequencing. I should have mentioned that Ashlea got that award, that recent award, which is just terrific.
Okay, thanks for that, Luke. Good morning, everyone. I'm glad to see that the majority of you know what the green tick is. I'll be talking a little bit about that shortly. And as Luke has mentioned, I'll be talking to you today about the research project I'm undertaking, Getting the Dirt on Nitrogen Fixation. This project has been made possible by the Department of Agriculture and Water Resources Science and Innovation Award for Young People in Agriculture, Fisheries and Forestry. I was one of the 14 lucky award recipients earlier this year, and my specific award was sponsored by AgriFutures Australia. This project aims to develop a fast, portable, cost-effective test that can be used in the field in real time to inform growers of what is happening in their legumes. We are currently halfway through the 12-month project, so I'll be going over what we've achieved and what we still hope to achieve. So many of you might have attended the earliest SNOC seminar about the Australian Inoculants Research Group, or AG as we affectionately call ourselves, given by my colleague Jessica Rigg. So I'll just give you a brief overview of AG and the quality assurance work that we do. A lot of our time is spent on these three key areas, all of which are focused on ensuring that the best possible inoculants are available to Australian legume growers. We curate a collection of commercial rhizobial strains, which we test annually to ensure they retain their efficacy. We endorse rhizobial inoculant products that have passed our stringent quality assurance testing as part of our green tick program, which is why we always recommend using products with our little green tick symbol on them that you saw earlier. We are also involved in a number of research projects in addition to the one that I'm discussing today, all of which have a focus on bridging the gaps between science and the field. Biological nitrogen fixation in agriculture relies on very specialized symbiosis between bacteria known as rhizobia and legume plants. Legumes include pulses like chickpeas and lentils, as well as pasture legumes such as lucerne and clovers, as well as the bicerula seeds you see here. Along with these seeds, you can see an agar plate of the corresponding rhizobial strain. And successful inoculation of legumes with rhizobia results in nodules on the roots of the host plant and also nice healthy plants. Here you can see some bicerula plants, and the large healthy looking ones on the left have been inoculated, and the sad little yellow ones on the right have had no inoculation. This symbiosis involves the rhizobia fixing nitrogen from the air and then supplying bioavailable nitrogen to the plant in exchange for other nutrients, namely complex carbon sources. Successfully nodulated legumes significantly reduce the amount of inorganic nitrogen required, as well as have increased yields and increase the nitrogen in the soil for following crops. If all of Australia's agricultural legumes were fixing nitrogen optimally, it has been estimated that this would save Australian growers $4 billion in inorganic nitrogen application annually. In reality, however, there are many variables that limit the actual efficiency of nitrogen fixation in the field. There are a number of these variables that we can control. The easiest of these is to ensure that you have the correct strain of inoculant for the legume you are planting, as having the correct strain occupying the nodules of your legumes will ensure the best potential for nitrogen fixation. It is also very important to store and apply your inoculant products as per the manufacturer's instructions, as rhizobia can be quite delicate and very sensitive to things like high temperatures and agricultural chemicals. Inoculant formulation and application methods might contribute to reduced nitrogen fixation, especially when in conjunction with poor soil conditions. Now, while soil conditions are controllable in theory, this seems to be one of the key areas that often fall short when looking at limitations to nitrogen fixation. Both the legume plants themselves, as well as their little rhizobia buddies, are very sensitive to a number of soil conditions, especially low pH. So it is important to either ameliorate poor soils or avoid planting legumes in incompatible soils. Plant cultivar is generally a small consideration, but is also something good to keep in mind. On the other hand, there are some variables that reduce nitrogen fixation that are completely out of our control, the current drought being the most obvious and troublesome one. In addition to climate, however, another thing that we might not always consider is competing rhizobial strains. So by competing strains, I mean strains that you are not inoculating with. These may be carried over from a previous planting, or they may be a native population. These organisms have the ability to nodulate the host you are planting and take valuable resources from your plants while supplying reduced amounts of nitrogen. We refer to these rhizobia as cheaters. And while there is no way to selectively remove these, if they are found to be occupying nodules instead of your intended strain, a further planting strategy could be devised to get around that. Now, while this symbiosis between the legume and rhizobia sounds lovely and friendly, that's not exactly the case. In nature, everything is a competition and everyone is looking out for themselves. In order to form this symbiotic relationship, both the plant and the rhizobia need to make and secrete chemical compounds in order to find each other and then to form a nodule. This process is especially expensive, as well as the ongoing nutrient exchange between both the plant and the rhizobia. This makes the whole process vulnerable to cheating rhizobia I mentioned previously. Australian commercial strains of rhizobia are the best available nitrogen fixers for their specified host legume, which is why the Ag team confirms the purity and efficacy of each commercial strain regularly. So, do you know what strain is in your nodules? Here we have some examples of the commercial strains and their specified legume hosts. This interaction is highly specific, to the point where some strains can only nodulate one host, while others that have the ability to nodulate multiple hosts can become cheaters in their non-specific hosts by supplying less nitrogen to those. This is why it's so important to apply the correct strain to ensure ideal nodule occupancy. So do we know what strains are in your nodules? Well, kind of, most of the time. Isolating rhizobia from nodules can be a laborious process and can take several weeks. Here you can see part of the isolation process, which involves several surface sterilization steps and dissection of the nodules before plating them on agar. And here is an example of the resulting agar plates we get after incubating them for some time. As you can see, we do not always get a clean isolate. And while this is not unexpected from environmental samples, it definitely complicates the isolation and purification process. Then, identifying isolates from environmental samples down to the strain level can be difficult. There are 41 commercial strains, most of which fall into 4 main genera. Many of the strains within these genera are so closely related that they are considered to be the same species, but they must have key genetic differences that make them behave differently with different hosts. Ideally, we want to design fast, accurate, and strain-specific molecular tools that remove the need to grow up and purify isolates from nodules, and also reduce time and costs involved in determining nodule occupancy in the field. To do this, we need more accurate whole genome information for each of our strains. So this slightly complex figure shows how related each of the different genera are to each other. As you can see, there are quite a few strains that fall into each of the 4 main genera, And then there's the one black sheep down the bottom, which is the only strain that doesn't fall into those 4 genera. Generally, standard molecular identification techniques, such as 16S PCR sequencing, can easily tell us which of these genera an isolate would fall into, but often cannot tell them apart any further than that. This is problematic, as some of these are so very specific. For example, these 3 Mesorhizobium cultures are so closely related that they are the same species, but have crucial genetic differences. That means they will only form nodules and fix nitrogen with one specific host each. These just barely different strains are known as biovars. Then there's this group of rhizobium strains. They are also the same species, but slightly different biovars, each with specific hosts. However, unlike the previous example, these strains can cross-nodulate different hosts, but when they do so, they do not provide as much nitrogen and become cheaters. In addition to these, most of the Brady rhizobium strains cannot be identified using standard molecular techniques. These are the commercial strains tested and supplied by Ag, with the highlighted strains here the most common, which is why we have chosen to focus on them for this project. Now, this is just to reiterate the relatedness of some of our isolates. Here is an example of the results we get from standard molecular tests, namely sequencing the 16S gene. As you can see here, the sequence that we submitted to the NCBI database search is 100% identical to many different strains of Brady rhizobium. In fact, this list continues for over 100 strains. This is where this project comes in. Here is a summary of the Getting the Dirt on Nitrogen Fixation project. We are performing whole genome sequencing on 21 of the most commonly used strains, which I indicated earlier, and we'll use that information to design strain-specific molecular assays. Now, since I'm discussing bacterial genomes quite a bit, I thought I would just give a brief overview of what I'm actually talking about. So the genome of an organism comprises all of its DNA, or genetic code. While these are pretty complex, bacteria are considered to have very simple genomes compared to plants and animals. Bacteria have one single large circular chromosome. In addition to this single chromosome, they also can and often do have plasmids, which are small compared to the chromosome. These plasmids are expensive for the bacteria to maintain, and bacteria have the ability to expel unnecessary plasmids. As such, plasmids that are maintained are usually very important for the survival of the bacteria. Nodulation and nitrogen fixation genes are found on plasmids. Now, there have been quite a few technological improvements in both genomics and qPCR that are making these technologies more accessible and have made this project possible. For example, the Human Genome Project that started in 1990 cost almost $3 billion, took 13 years, and was a mixture or a mosaic of samples, not just one person's whole genome. Whereas today, you can send off a swab in the mail and have your own personal genome sequenced for less than $1,000 in a matter of weeks. And then, while some versions of thermal cyclone machines, which are required to run the molecular assays we want to design, have been designed to be portable, making it possible to perform these molecular assays in the field. Here you can see a comparison of commonly used thermal cycler on the left, which can be found in many molecular labs, compared to the MIC thermal cycler on the right, which I'll touch on more shortly. We are using a combination of short and long read sequencing technologies, which have their own pros and cons, as you'll see here. The first sequencing technology we are using is the Minion. It's an impressively tiny sequencing device made by Oxford Nanopore Technologies. As well as being tiny, it is also very cheap at just $1,000. However, much like office printers and ink these days, while the device is cheap, the consumables are where all your money goes. The flow cells, you can see one sitting in the device here, are $900 each. However, Oxford Nanopore are continuously improving the chemistry of their flow cells, so you can actually get a lot more data from a single one now than you could even a year ago. The key advantage of the Minion is that it sequences very long lengths of DNA, many thousands of base pairs in length. The numbers here are what we have achieved in the lab so far, though the maximum reported read length to date is over 2 million base pairs. However, the one main issue with long-read sequencing is that it has a relatively high error rate at approximately 5%, meaning that around 95% of the bases we get are correct. The second sequencing technology we are using is the iron torrent by Thermo Fisher. This iron torrent while the iron torrent is not as small nor anywhere near as cheap as the Minion, it has two key attributes that made us choose this specific technology. The main reason is that the iron torrent uses short read sequencing technology, which has a much lower error rate of about 1%, meaning that we are able to use these more accurate reads to polish or correct errors in the long reads. The second and possibly equally as important reason we chose to use the iron torrent is that we have access to one here at the Elizabeth MacArthur Agricultural Institute, which means we are able to save money by avoiding outsourcing. It also gives myself and the ag team a chance to learn new valuable skills. Now, we are using these two different sequencing technologies in order to make the best of both worlds. Long reads from the Minion cover large sections of the genome and can result in complete genome sequences, but as I mentioned, there are likely to be a number of errors. Whereas the short read sequences are more accurate, but often result in gaps in areas of the genome where there are lots of repetitive sequence. By assembling the long read sequences, we essentially make a map or a scaffold of where the short reads go in relation to these gaps. We can then use these more accurate short reads to correct the errors in the long reads, and this will hopefully result in whole accurate genomes. So far, all but one of the 21 genomes have been sequenced using the Minion, which has resulted in about 1 terabyte of data, which is a lot for your average computer to deal with. As such, we have enlisted the help of one of the bioinformatics experts on site at EMAI in order to process all of the data and assemble the genomes in a timely manner. Here you can see two different visualizations of the long read assembly results of the TA1 strain, which is specific to white clover. These will then be polished with the short reads from the iron torrent. Once sequencing is completed and high quality genomes are assembled, we will line all of the genome sequences up side by side and compare them to find unique parts of their genomes. The fact that each strain interacts differently with individual hosts suggests that each strain should have something just a little bit different about their genome. These unique regions of each genome will be our targets for designing molecular assays. We will then assess and optimize the performance of those assays, and multiplexing allows us to cut down on consumables and time by combining multiple assays in the same tube. Hopefully, we will find a number of regions unique to each strain so that we have several chances at finding winning combinations. Using another technological advance from recent years, we will be able to perform the assays in real time in the field. As I showed earlier, most thermal cycler machines are large and cannot be moved. In contrast, the MIC has been designed specifically to be portable. It is small, efficient, and compatible with both car batteries and solar panels. It also allows us to multiplex up to 4 different assays in a single tube, increasing capacity. Using this machine in conjunction with our assays, we will be able to dig up a plant, sample the nodules, perform the assay, and have an answer in a few hours. Once we have the assays optimized and working with the nodules that we have generated in the lab, we plan to run field trials at DPI research sites and also possibly at pulse, legume, or pasture workshops and meetings. Some of the practical applications we hope this project will have are in answering some key questions regarding inoculation, such as, was inoculation successful? Could other methods possibly be better for your situation? Do you need to inoculate more than recommended? This could be the case if there are competing rhizobia present or if soil conditions are suboptimal. How much does soil type impact the success of nodulation, as well as how well do new cultivars nodulate with current strains? In addition to the knowledge and insights, we hope the assays that result from this project will bring to current and future field research. The genomic data we are gathering will provide a wealth of information for future research projects. So hopefully I've convinced you of the importance of nodule occupancy, and watch this space for updates on the project outcomes. I'd like to thank AgriFutures Australia and the Science and Innovation Awards, as well as New South Wales DPI and the AG team for making this project possible. And thanks to Luke and Abby for giving me the opportunity to speak today. And last but not least, happy World Soils Day, everybody.
Well said, Ashley. Thanks for the reminder about World Soils Day. And thanks for going back to that slide. Yeah, absolutely terrific, Ashley, and very nice and concise, very exciting work coming up there. So now we have the opportunity to take some questions. So I might go to Abby.
Yes, thanks, Luke, and thanks, Ashley. That was really great. I wondered, are the assays that you've talked about specific for legume nodules? Did you want to comment?
Sure. So they're not specific to just legume nodule samples. In theory, these assays could be used on any sample type, including soil, which means in the future there might even be the possibility of checking whether the correct strain of rhizobia is still around in your soil from previous plantings. However, there will be some significant limitations to this application. As soil is a very complex sample, we would not be able to run the assays in the field. We would have to bring the soil samples to the lab and process them beforehand. There is also the limitation of being able to accurately quantify the amount of rhizobia in that soil. So just because you know that you have some of that strain there, you wouldn't necessarily be able to accurately know if there's enough there to get the most out of your legumes. So even if we were to do that, we would still always recommend to inoculate again to make sure that you've got the best options.
Okay, thanks very much.
Now, we do have a question here that I've had from John Paulet, and John has asked, what are your thoughts on the value of inoculation with alternative fertilisers / compost teas, etc.? Are there known benefits or downsides? Thanks, John.
Thanks, John. Good question. Currently, we haven't actually had the opportunity to assess the effectiveness or efficiency of rhizobia when inoculated with some of these other non-rhizobial inoculants. There's certainly some papers that indicate it could be beneficial, but we don't have any solid evidence at the moment to say that they are always beneficial in every situation. And unless you actually know exactly what is in some of those teas, it would be hard to say at all whether they would be beneficial.
And I can't see any other questions there. I just had one quick one for you, Ashley, which was, how do you know that the commercial strains are the most effective ones?
Sure. So the short answer is that there's no way to know if we have the best possible strains ever, as evolution is constantly happening, but the commercial strains are the ones that have been proven to be the best that are available. So in order for a rhizobia strain to be part of the list of commercial strains that we have, whether it's a completely new one for a new crop, or if it's to replace an older strain, it must go through a vigorous assessment process requiring multiple field trials across multiple seasons and multiple soil types. And the results of these field trials then undergo a peer review process by a panel of experts who then vote as to whether or not the data shows an improvement over the previous inoculant. So while there's certainly always room to potentially improve those strains, they're the best we have evidence of at the moment.
Good answer. Arnie Vankov's got a couple here.
I was just going to ask Ashley, some of those strains are the same species. So I was just wondering if she's actually had a look at, I know for a fact that some of the species have been sequenced, and whether she's compared the differences of those that have been sequenced and whether those differences are actually genome located or plasmid located.
So that's something that we'll definitely be getting into once we have some of our own genome sequences. We'll definitely be comparing all of the available sequences to make sure that we don't have any cross reactions with our assays. I haven't as yet, but one of the reasons that we have decided to go ahead and sequence our some of those ones again that we have in our collection is that they have been separated from the original collections they came from, which is where I assume the sequences that are available have come from. They've been separated for decades potentially, and we don't actually know what's happened to either of those in between. So there's always the potential that even though we have the same strain, there may have been some change either in our library over the decades or in the original library over the decades. So hopefully we'll be able to tell that.
I can just see strong parallels to cheese starters and yogurt starters. They were basically the same species, different they weren't even different biovars, but what they did have were huge, a variety of different plasmids, and some of them were quite small, some of them were quite large, and we spent, I think the group that I worked for spent nearly 20 years, and they came up with the theory that all the differences were basically due to the plasmids, because bacteria are quite promiscuous. So yeah, that's all I wanted to say.
Yes, I wouldn't be surprised if a lot of those strains that are the same species do have the same, do have identical chromosomes and just have slight differences in the plasmids. But even that will be very valuable and give us at least something to target with those.
Yep. Good.
Thanks, Tony. I can't see at this point any other questions. Looks like, Ashlea, you have covered everything.
Great.
Perfectly. So, yeah, very informative, really interesting, and good luck with the work, and congratulations again on being so well recognised. It's terrific.
Thank you.
Thank you very much for your support and listening in 2019 on behalf of Abby and myself. It's been a pleasure bringing such terrific talks such as yours today, Ashlea, to you all, and we look forward to seeing you all in the new year. So happy Soils Day and Merry Christmas, and we'll talk to you all next year. All right, well, I'll call that a close. Thanks again, Ashlea. We'll talk to you all next year.
Bye, everyone.
Bye.